Alright, ladies and gentlemen, this is going to be the next video in biology, specifically going through and discussing translation. Let's get started on this next video. So if we're going back to the overview here, we've already discussed transcription, and now we are here. We're discussing this bottom half here, translation, where we're going from our RNA into our protein. Alright, so what specifically is translation when we're going through and thinking about this? Well, translation is going to be the second part of that central dogma, what we're going through and learning. We're going from RNA into protein, and it's the process in which the genetic code in mRNA is read. Now we're going to talk about codons and it's being read at one codon at a time. Now there are three main steps when going through and discussing this. We have the same as transcription. We have initiation, elongation, and termination. These are the pieces and the processes that we're going through and looking at. Now there are also going to be players in translation, which we went through and discussed before. We have mRNA, the ribosome, our tRNA, and the amino acids which are attached to our tRNA. Now if we're going through and thinking about, well, where does this take place? Well, it's going to take place either in the ribosomes along the rough endoplasmic reticulum, or it's going to take place in the cytoplasm, in the cytoplasmic fluid, again in a free-floating ribosome. Now there's a concept that we have to understand here. We have to understand codons. Codon is the region on the mRNA, our messenger RNA, which is 3 nucleotides in length, that codes for a single amino acid. And then we have to understand anticodons. Anticodons are 3 nucleotide sequences located along our tRNA, our transfer RNA, and they're going to correspond to the appropriate codon along our mRNA. So we have a codon here and we have an anticodon. And what we're going to do is we're going to send our mRNA sequence into our ribosome, and the codon is going to be read and matched with the anticodon to get the appropriate amino acid sequence. So let's go through this process in detail here. Let's start with initiation. Initiation is the beginning where we have our 2 subunits, our A and B subunits of our ribosome that are going to come together. Now we have 3 sites. We have our E, P, and A site here. Now the A site is going to accept incoming tRNA. The P site is going to hold that tRNA, and then we're going to see the E site where they go through and exit. So what we're going to initially see is our mRNA sequence is going to be bound to that ribosome there. We have our codons along that mRNA sequence. And when we go through and discuss elongation here, what we're going to go through and see is our tRNA are going to be accepted, and then we're going to match up our corresponding tRNA with our mRNA. So we have the first start codon here, AUG. The appropriate tRNA codon here is UAC. What we're going to see is these tRNA are going to bring the appropriate amino acid, and what we're going to see is our anticodons, and our codons are going to line up along these sequences in order to get the correct amino acid and polypeptide sequence. So we're going to see that the next tRNA is going to move in, and what we're going to do is create a mechanism, a machine that is going to go through and allow for the next tRNA to move into our accepting site, and the previous tRNA to go through and exit. And what we see is over time, this is going to go through and create a chain, a polypeptide chain of amino acids, which is going to go through and allow us to get the appropriate folded protein. Now with the end of this, we're going to see that we're going to have this polypeptide chain, and eventually our mRNA sequence is going to go through and end. This is the last step here where we get to termination. Termination is where we're going to see our tRNA exit, mRNA is going to exit, our ribosome is then going to decouple, and we're finally left with our protein. So let's go through and break this sequence down into a couple of different steps here. What we're going to see is the first step here. This is the initiation step. In the initiation step, the ribosome is assembling around the mRNA to be read, and the first tRNA matches with the start codon AUG. So the A and B ribosomal complexes unite. We see the mRNA bind to the ribosome, and we see the initial tRNA go through and bind. The second step here is elongation. Now in elongation, this is where the amino acid chain gets longer and we go through and build that polypeptide. Each time a new codon is exposed, we're going to see a couple of things. The matching tRNA is going to go through and bind to that codon, and the existing amino acid chain is linked onto the amino acid of the tRNA. Now lastly here, we're going to see our termination. This is the stage in which the finished polypeptide chain is released, and there are 3 stop codons associated with this, which we'll learn about in a little bit. Now, the tRNA is going to be released. We're also going to see the polypeptide chain be released. And the polypeptide chain is going to then be folded via another organelle within the cell. Remember, there are different ways we can fold up these polypeptide sequences to get the appropriate 3-dimensional structure. So let's go through and look at this mechanism again from the beginning. In the initiation sequence here, we're going to have our A and B subunits come together, and our mRNA is going to be accepted by the ribosome. Now at the end of the initiation sequence, or we could say at the beginning of the elongation sequence, what we're going to see is that our initial tRNA be accepted by that initial start codon. As we go through this process, remember the tRNA, the transfer RNA, is carrying the anticodon, which is going to match up with the codon along our mRNA, and it's carrying the appropriate amino acid. As the next tRNA gets accepted, we're going to go through and then create our polypeptide chain according to our sequence of tRNA anticodons with the codons along the mRNA. And as we go through and move through this mechanism, we can see our polypeptide chain is going to grow and get longer. At the end of this sequence, we're going to hit one of our 3 stop codons, and what that's going to signal is our tRNA and our mRNA to decouple, and we're going to be left with our final product, our protein. So what is going to be the end result of translation? Well, at the end of this, the product of translation is our protein. We're trying to go through and get our protein sequence in that 3-dimensional sequence that we're going to go through and fold up later, that's going to go through and be this solid structure or provide enzymatic functions somewhere in the body. So the product is a protein / amino acid sequence that is later used downstream for the cell and the organism. So we are going from, in this process, RNA into our final product, a protein. All right, so there was a lot in that video. Did you learn a couple of things? Did you learn what translation is? Did you learn about the 3 steps of translation? And then lastly here, what is the end result of translation? This is going to be the end of the video. I will see you all in class tomorrow.